Please introduce the webinar.
Hello, everybody. Dear members of the ISCBTT, dear colleagues, I want, as president of the ISCBTT, I wanted to introduce this webinar because it's a major event of our society this year. You know what the reasons are, in particular the postponement of the Fourth World Congress to October next year. We hope that everybody will be in Copenhagen next year for a successful and very interesting World Congress. The motivations of the Council to organize this webinar, and I would like to thank the Council, of course, in this slide. Is where the motivations of the Council were to preserve the link between the ISCBTT members, in spite of the fact that we don't meet all together physically in Copenhagen this year, and also to organize an event around the General Assembly that will be held tomorrow, and I hope that you will be all there because we have very important discussions to have during this General Assembly. I would like to thank all the inputs of the ISCBTT Council. The members are listed on the left side of this screen, but also I would like really to thank the organizing committee of this webinar that was led by Julie Gill. Thank you very much, Julie Gill, for all your efforts in organizing this webinar. And the other members of the organizing committee were Richard Heller, Javier Raso, Rafael Davalos, Giovanna Ferrari, Eugene Boroviev, Giampiero Bataro, Nabil Gremi, and myself. With this, I pass the word to the chair of this first webinar, Javier Raso. Javier.
Hello, everybody. Good morning for those that are in the American time zone. Good afternoon for those that are in the European and African time zone, and good evening or good night for those that are in the Asian and Oceanian time zone. This is Javier Raso. I am professor of food technology at the University of Zaragoza in Spain, and among my colleague Giampiero Pantaro, professor at the University of Salerno in Italy, we are going to moderate this webinar. First of all, Giampiero and me will like to give all of you a warm welcome to the webinar organized by the International Society of Electroporation-Based Technologies and Treatments. 2 years ago, when the successful third edition of the World Congress held in Toulouse ended, everyone expected at this time to be in the beautiful city of Copenhagen for a new edition of the Congress. However, as everybody knows, unfortunately, due to the uncertainty that we are living in these troubled times, the organizing committee of the Fourth World Congress, with the approval of the ISBTT Council, decided to postpone the fourth edition to 2022. So we are today here in front of our computer because this year it has been decided to organize 2 webinars prior to the General Assembly of our society. I would like to remark that this webinar don't try to replace the World Congress, as it is indicated in the web page. It's just an appetizer for the next year World Congress. The main objective, as Luis said, of this appetizer is to maintain the contact between the members of our society. I'm going to share my screen in order to show you the program, the today webinar. Okay, so the webinar consists in 3 parts with 2 small breaks between each part. The first part is devoted to food technology, and we will start with a presentation by Professor Indrawati OA on an overview of non-standard applications of pulse electric fields in the food industry. After Indrawati's presentation, we will have 2 presentations on non-conventional applications of pulse electric fields in biotechnology for inducing factor release in Chlorella vulgaris and for production of metabolites from lactic acid bacteria. After the first break, we will follow with the second part of the webinar that is devoted to basic science. We will start with a presentation of Professor Antoni Iborra on avoiding nerve impulse in and by electroporation. And then we will have 2 presentations by Siki Wu and Log Jibot about their research on nanoseconds electric pulses in immunosuppression in tumor microenvironment and effect of cold atmospheric plasma on the stimulation of dermal collagen remodeling at tissue scale. After the second break, we will finish the webinar with a talk by Professor Pierre Jais on the effect of pulse electric fields in cardiac ablation. Before finishing my presentation, I would like to give some warnings from the organization. The first warning is concerning questions to the speakers, Zoom platform, and keep your microphone mute. Moderators will forward the questions to the speakers at the end of the presentations. The second warning is that the talks are going to be recorded and they will be available for attendance on the web page of the International Society of Electroporation-Based Technologies and Treatment. And the last warning, perhaps the more important, I encourage all of you, all the members of the society, to attend the General Assembly tomorrow at 15:30 Copenhagen time. Several issues of great importance for the future of the society are going to be discussed. I remind you that it is possible last-minute registration for the members of the society. So you are on time of register to attend the General Assembly. I would like to finish in thanking all of you for joining us to this webinar from your home. I understand that it is always less attractive to participate in this kind of events online than physically. So I look forward to meeting all of you in 2022 in Copenhagen for the 4th edition of the World Congress. I know that the organization committee has made a big effort to prepare for all of us a great cross-disciplinary event in order to overcome or at least to match the successful previous editions of the World Congress. So in this sense, I remind you that the submission system is open for accepting short abstracts for the 2022 Congress. So thank you very much for your attention, and I pass the ground to my co-moderator, Giampiero Pataro, for introducing the first speaker of the webinar.
Hello to everybody, and thank you, Javier, for also introducing me, and welcome to everyone to join to this first webinar. According to the program that Javier showed you in detail before, we will start now with the first 3 presentations in the frame of food technology. And to this regard, it's a pleasure for me to introduce the first speaker that, although now in New Zealand is middle of night, she's here with us to present us an interesting applications, alternative applications of PEF. She's Professor Indrawati Oei. Who is she's the head of food science department at the University of Otago and principal investigator at the Reed Institute of New Zealand. Please, you can probably switch on the camera. Okay, thank you. You know very well Professor Ray, she has a large experience in post-harvest innovation and novel technologies, including, of course, pulse electric field. And we are all very curious to follow her presentation, her lecture, on, in which she will provide us an overview on non-standard applications of pulse electric field. Please, Idrawati, the microphone is yours.
Thank you, Mr. Chairman, for introducing me to this conference, and thank you for inviting me to give a talk and share our experience here in New Zealand. Good morning, everyone. Good afternoon, good evening, and greetings from New Zealand. I'm in the midnight time here. So when I received the invitations from the organizations, that I'm very excited with the title about the non-standard food applications of PEF. So I'm trying to go back to my first slide. Yeah. So this presentation has been prepared with my colleagues also, Xiying Leong from the University of Otago in New Zealand. And also I see a lot of my students and my PhD students is also attending this conference. So thank you very much to joining this webinar, because I would like to share also your research in this webinar. So first of all, when I get this title from the organizer, I'm asking myself about why they give me the title so quite unique, non-standard applications of PEF. So as we know that not many food products are homogeneous, while we know already in the theory and a lot of books that we learn that normally PEF should be, you know, a lot of requirement around homogeneity about matrices. We need to be very pragmatic about the size. We need to make sure that everyone understand that the electric field strength has to be delivered in a homogeneous way. However, we need to understand when we apply PEF to the food applications that foods cannot be homogeneous. So foods always in a different system and very complex. Sometimes it's solid, inconsistent in dimension, heterogeneous, and sometimes also multi-layers. So for example, like in this case, onion, even though it is, people use it quite a lot as a sample, but people forget that basically onion is also very complex matrix. And we also talk quite a lot in the literatures around the fine particles if you are working with flour, and also in the viscous matrices. So because of that, why our research at Otago or our colleagues do in the different institutions is quite interesting about how we can actually apply PEF for food materials. And I think a lot of my colleagues now also call us as a non-standard food applications. Because if you say here, that is the reality that we will have. So if you see the PEF, if you are following the theory, this theory does not work with the food system. Because if you see here, you can have like orchard tubers. People always say, yes, this part is actually homogeneous, but in the reality, if you see carefully in the microscope, it is not uniform. And also depending on the positions between the electrodes, that also give you also influence in the directions in the current, how we can deliver the current and also electric fields. And when you do the multi-layer, sometimes we need to think how actually the directions of the flow from the electric current too. So because of that, we also have seen heterogeneous impact in the food matrix, and also how is actually the applications afterwards and what is the good impact that can be resulted. So this is part of the trial that we use plant materials, and we also use animal based type of food. So in this left side here, people always talk, hey, PEF can be used for the low value type of meat. Please use this technology because you can make tenderizations. But in the reality, if you want to go in the food applications in the industry, that is the low value meat. So you have a different composition of fat, different connective tissue, and also the muscle directions. It is really have a different directions, even in one cut. So for example, like in the seafood, yeah, so this is pawa. In this product that you can see that the muscle directions is also different. Yeah, so you can see also non-homogeneity in the matrix. So basically, if you see in this topic, people always say, yeah, there is no application at all for PEF. But because of that, it challenged us about our curiosity, and I would like to know more whether it is really not possible. Because what we need to see is it's about discovery. So discovery of new technological application of PEF. And I would like to share with you with the 5 case study that we have been working on for years. There is quite an opening application that might be given by a pulse electric field processing. The second application is also to tap into the underutilized raw material. So at this moment, a lot of extractions procedure we just discussed for the premium product, but we never seen about more complex product or about the waste that is coming in the industry. The second driver why also we need to explore in the non-standard food applications about by 2030, we need to go to sustainability missions goal that we want to achieve more efficiency in the productivity, less energy, and as also we can use all of the metrics that we provide or we produce in the farm. So with this type of questions that can we do a new product that it is coming with the PEF because it's never been applied so far. So all of the applications so far from PEF, it is around pasteurization, around liquid. Everyone knows about that applications. We also have a great success in the grape industry for wine maceration, wine productions for the maceration of grapes, and also for the potato industry. So that is a 3 key application that people always think it is as a gold standard from PEF. However, when we do from the academic point of view, working with the industry, sometimes we need to find a niche of these applications. Especially we understand food is so complex, heterogeneous in the matrix, and sometimes in the inconsistent dimension. So I will give you also the challenge here. A lot of research that we do sometimes has been criticized by the experts that is working in the standard food applications around the energy and electric field strength that we use. So just to bear you in mind that if you are working in these applications, that we are not talking about one cell. We are talking about a small piece of tissue. What we are talking about the bulk of the whole organ of the plant or the whole chunk of meat. So that is also, hopefully with these presentations, we can have also more discussions in the future because we have been challenged by chamber design and also the capacity of generator, how we can actually deliver the processing is more efficiently. But of course, all of the justifications is also depending on about the food safety, and we are trying to do that as less as possible impact in the sensory attributes. But hopefully by learning from the functionality, everyone can see also how we can actually combat the challenge when we designing the chamber design and also the generator. So our first study that I want to share with you around animal muscle tissue. So a lot of our past study that we found in the literature is always coming from the perfect steak type of cut. But in the other side, in the review, always people say, hey, we need to use for the low value meat. But this is low value meat, that is in the reality. So with the reality, the challenge that is quite clear that it is uneven distributions of muscle fiber directions. You have also fat. We have a lot of connective tissue and collagens. And also we see that the electrical conductivity across the cut is a very variable, and it is very different from cut to cut and one animal to the others. And sometimes it is a very big jump from one cut. It can be 3.2 millisiemens per centimeter until 9.4. So that is, I think, I want to share you the challenge here when we are working with the food product in the industrial scale. And also the presence of bones sometimes can also hinder the process, or also sometimes can change the directions of the electric current. So here that more and more we are working with the solid matrix. We have also challenge in the treatment chamber that can suit for different cut, for different dimension. So to have an upscaling for continuous process at this moment is still limited. So we are hoping that there is an advancement within coming few years so that these applications can be applied. So I just want to share what is the good things that why can we use PEF to increase the value from the low cut. So we found that the PEF can reduce the denaturation temperature of the connective tissue. So when we increase the specific energy, that basically we can have more, lowering the denaturation enthalpy. So what does it mean by that? That if we do PEF as a pre-treatment before any thermal treatment afterwards, that we can reduce the intensity of the thermal treatment subsequently after PEF treatment. So this is also we found that in this image that before PEF, you still see that the solubility of the collagen's fiber can be increased after PEF. So what is the impact then, and what is the application? So PEF basically can precondition the low value soft meat. And one is the good applications that we also learn in the FIAT project that is funded by New Zealand MB Ministry of Business and Employment, that we can increase the value of the low value meat. So in this case, PEF treatment can modify the microstructures of both microfibria and connective tissue, and because of change in the thermal properties of the collagens. So commercially, you need very high temperature to sous vide meat to get a certain texture. You need 1 until 2 days, but with the PEF treated biscuit, you can shorten again the sous vide time, half of the time, and even lower temperature at the 60 degrees. But the beauty of this processing that you can have more tender, and also the texture is more like steak, like type of quality. So it is quite a big improvement coming from the animal productions. And the beauty also with the PEF, maybe a lot of article has been published maybe around year 2000 when we start with the PEF research, or maybe year 1998. That people has like assumptions or hypotheses that PEF improve the protein digestibility or digestibility of macromolecules. So in our trial, we also found that PEF basically, indeed, you can also improve the protein digestibility of sous vide cooked meat. So that is the, if you can imagine the in vitro digestions, it is from the iron digesters and it is a PEF. So basically, after simulated digestions, that we can have more protein digested in the gut. So the second case study that I want to highlight is around shellfish. So the challenge with shellfish that is described as a soft body invertebrate that exists within hard calcium carbonate shell. So people always say, yeah, that is an insulator. How can you do it with a PEF? And also, when we remove the shell, there is a types of protein and muscle that is coming from a different animal. So it is totally different concept between animal muscle tissue and shellfish tissue. And the electrical conductivity is very high. As you know, that shellfish is actually living in the seawater. So the salt is actually increased the conductivity. And then for the upscaling, it is still limited because we need a new generator that we can also process that appropriately. So when we apply PEF basically in intact muscle, so for example, like in pawa, that we can see here that pawa or abalone, that you can basically reduce the hardness and the chewiness of cooked abalone with the PEF pre-treatment. So these applications, you can also apply for homogenized muscle, which is you can also create a colloid form, which is with the high intensity of field strength, we can assist the protein extractions, and you can improve the extraction yield by 77% of the muscle protein. And also, PEF can actually change the functionality of the isolated protein because it exhibits good emulsifying and also the solubility properties. So this enable the production of high protein products from the homogenized muscle. So the third applications is around grains and seed. So a lot of people, when I start the research around grains, our challenge basically, this is totally different than shellfish or animal-based tissue. So grains and seeds has a very low electrical conductivity. Yeah, and also when you see the structure of grains and seed, they are multi-layer. And then the layer, if you're coming from the other side, it is in the coat, they are actually insulator. Yeah, and then just coming inside and then coming in the sperm and then afterwards go to the seed. So the other challenge that we see is the hard structures of the grain and seed, but the anatomy and the compositions between species is totally different too. So for example, like even like wheat or corn seed or bean seed, they are in the structure is totally different. But the differences between, for example, like shellfish and also animal-based type of product, that grain and seed still possess a living system. So what does it mean by living system for plant? That grain and seed can still have a capability to grow. So with this concept of cell electroporation's impact, that PEF can be used to trigger the plant defense mechanisms. So in this case, we also have found, and then very interesting, that we can do seed electropriming with PEF. The question sometimes, why it is important? Because electropriming, this has been done, or priming is important when we grow seed. So normally we can do chemically, so abiotic and biotic stress that can induce basically antioxidants. But PEF is very interesting when we apply it to the seed. Basically, you are doing in the very green technology, low energy, and also clean. There is no additives that has been added. So this is fully physical processing that we give to seed. But the crucial things is to find the water content inside of the seed, that you need to imbibe the seed before the PEF treatment. And then by doing this, you can improve the growth of the seedling and also improve the high level of the production in the antioxidants. So it is more nutritious, it is more healthier. And we have already proved it in the Kakko cell that after the in vitro digestions, the PEF treated type of seed that we grow afterwards, the seedlings has a higher in the antioxidants, but also higher in the bioprotective activity. So the fourth case study that we also use quite a lot about the fine particles, powder and flour. So we have also challenge here. Again, the same story as like seed and legumes, that we need to precondition them, okay? And because it is dry and also low conductivity, and we need to use the suspension. And it is very critical here to understand that we need to have a proper dispersions before and during PEF. So with the current chamber design that we have, that is already available in the market, or people can design now, that sometimes we have a separations if we wait too long. So here that we need to have a system that we can make a suspension quite quickly, go to the treatment, and afterwards we do another treatment afterwards for maybe heat treatment or other applications. So here we always see that the native form in the biomacromolecules, in this case, like isolated protein and starch, is a very heat and pH sensitive. So we need to have a problem also in the monitoring the temperature during treatment, and it has become crucial. But again, we have a challenge because the system is not homogeneous in the chamber. So that is not our result. It is coming from our colleagues, that is Jiang et al., that they are trying to do the PEF applications directly on flour, so without any liquid preconditioning. So the rationale that the group has been using this type of approach, they want to avoid wheat gluten forming dough before and during the PEF itself. So what they found is quite interesting, because the wheat gluten that is coming from the PET pre-treatment, they can form more stable network of the structure is quite stable, but also the molecular shape of the gluten protein was not altered. So in this case, also we found similar like in the animal-based product, there is a reductions in the thermal stability, and also very interestingly, it is reductions in the hydrophobicity of the gluten protein. So what is happening? That is a new applications coming that PEF basically can enhance, for example, like start performance during the 3D printing, that in this case, by reducing the viscosity, crystallinity, and also gelatinizations and enthalpy, you can form firmer gel. So if you are applying this technique to the 3D printing, that we can have a smoother surface in the printed product. So you are using the same ingredients, but with the PEF treatment, that the products will be different. And also, quite importantly, that for 3D printing, that you have a low adhesiveness. So based on the results coming from our colleagues from the other group, that we also use now a new idea for the compatibility, that PEF can enhance the compatibility between protein and polysaccharide interactions to develop a stable biofilm. So in this case, the idea behind this, we have plant protein, zein, and chitosan, that is carbohydrate coming from the shellfish, and then we have make different emulsions. So by changing different pH, so one is in the conservative type of matrix, and the other one is with the non-associating complex, that you can enable productions of stable, eco-friendly, low-cost, and non-toxic type of, hopefully it is edible film to protect food products. So you can use it actually for the coating for fruit or cheese, for example. So with these applications, when we move again in the food, like what we always call it as the perfect situation is a homogeneous food, again, that I want to make you aware that even though you are working in the different metrics, for example, like water, milk, oil, syrup, heavy syrup, or chocolate syrup, you will have different viscosity. So it is a very critical here when we do this type of applications, that we need to have a proper dispersions of viscous food before and during PEF treatment. And that is a higher tendency of the product sometimes falling in the transfer line and also in the treatment chamber. And what we can see here, the viscosity changes with the temperatures and time, and pH, I'm sorry. So here that we also realized that the electrical conductivity varied between food type. So I will give you one example that is coming from Dr. Yafi Liu, that is also in the audience now, that he is working in the egg white type of solutions, that we study the impact of pH, and also we are comparing between heat treatment and PEF treatment. So in this case, what we found that with the heat treatment, the protein degradations create like a turbidity in the product. But when you do the PEF treatment, you can still achieve a clear egg-white solutions at the high pH here. And that is very important for us is around what, so what? If we are using this product in the, for example, like in a non-dairy drink, we can produce a clear and highly digestible high protein. So here that we are comparing about the control and the thermal and the PEF, then you can see having the same impact in the digestibility, but you will have a different here turbidity and also product. So here it's quite clear that we can have also non-turbid type of egg white after PEF. So what is the take-home message here? And I think call of everyone who is expert in the PEF and also from the engineering point of view, equipment design, and also from industry applications. Yes, there is a hope for non-standard applications for PEF, but at this moment, we still have a challenge by the chamber design to have the design in the generator and also how we can select the conducting medium, how we can fit the product actually inside the chamber to meet the food safety. And also we can have as minimal as possible the changings in the sensory attributes. But also importantly that it is, you need to see in the future that PEF applications on the heterogeneous food matrix with the complex structures is promising. And it is opening a lot of novel applications that we can use with the combinations of other technology. And for example, preconditioning the raw material for 3D printing, you can have a product in the nutritious seedling, high protein, bread making, and biodegradable edible food packaging. So by doing that, hopefully the industry update can maybe can be fastened. It is not as slow as now. So I would like to thank to all of the two founders of our research in the last 6 years. It's Sept from the Food Industry Enabling Technology funded by New Zealand Ministry of Business, Innovations and Employment, and also Research Institute, New Zealand Centre for Research and Excellence funded also by New Zealand Tertiary Education Commissions. I thank to all of my colleagues that is listed here, Professor Phil Bremer, Pat Silcock, David Buret, SM Ali, Alan Karn, Dominic Akie, Siying Leong, that is also co-author of these presentations, Yafe Liu, that is also attending at this moment, Stephen Kitero, Amali, Seba Marby, Alpos, Nigel Narson, Jaspreet Singh, Mike Boland, Lofty Kong, Feng Ning Chiang, and also Nasima Hamed. So thank you all for your attention. If you have any questions, please feel free to ask. Thank you.
Thank you, Professor Ray, for this very interesting overview of different applications, non-standard applications of PEF on different materials from meat, fish, powders, and so forth. So I would invite all the audience if they have some questions, and I also invite you for the next presentation to remember to type your questions in the chat of Zoom. But in this case, we can proceed with giving you the microphone if you have questions. Curiosity. Okay.
Please take the questions in the Q&A.
We have one question in the chat.
I can see here the questions from Nacho. So nice. Thank you for your questions, Ignacio. Thank you. Yeah, that is one of the challenges that maybe some colleagues can help, because we have also comment from the reviewer. They always, surprised with the high energy that we use, especially with the solid materials. At this point, because of the capacity of the generator that we have, we need to create very high energy to see that functionality. So hopefully, if we have more funding, we can have better generator, and hopefully we can learn more how we can optimize the delivery of the energy and also the current. So to be honest, I cannot answer your questions, but that is hopefully I can have better generator or higher generator that can cope, because we know already the challenges that we have. I see also questions here from...
Yeah, from we have other two questions, if you can...
Yeah, I just want to make sure that I understood the questions, because from Borkos, collections on so-called blue food in nature, referring to all food sourced from aquatic environments, points to a growing demand in the world and growing possibility of providing nutrition of the growing world populations. Given that they say that the majority of the producer of the blue foods are small, with possibly limited access to advanced machinery and funding, how do you see the role of PEF in the development of this possibly very sustainable source of nutrition? So thank you for your questions. So I have also the same question for myself, because New Zealand is small, and we have been challenged also with the quantity of the productions, how we can make it efficient, the applications of this technology, and also make sure we have enough funding to purchase the equipment. So I think this call should come to the equipment designer or also the equipment company. So hopefully that they can create a machine that can produce with the low cost, and then also with the different size or scale, so that the company does not have to invest as high as at this moment, the cost that they need to spend. So that is maybe the only solutions to make it sustainable. And the smaller the equipment that we can also movable from one farm to the other farm, it is will be more sustainable in the long future. But what I want to share here is about the functionality and then the potential, so can create an opportunity for the industry to uptake afterwards.
Okay, thank you very much. I think now the topic, of course, is very interesting, but probably we have to move on with the program of today. So I thank you again, Professor Ray, for this very interesting talk. And we move to the second speaker, who is Damaris Krast. She's has a background in chemical biology and now is a PhD candidate at the Karlsruhe Institute of Technology, German, where she's working on a topic focused on biological response of microalgae to PEF treatment for improvement of protein extractions. And today, in fact, her talk is titled Cell Death Induced the Factor Released by Pulse Electric Field Treatment in Chlorella vulgaris. Damaris, can you please switch on the camera and the microphone?
Yes.
Okay.
So you should be able to see now my screen.
Yeah, when you want, you can start, please.
Thank you for the kind introduction and the opportunity to speak for this webinar. Hello, dear participants. My name is Damaris Krast, as I was already introduced, and I'm a PhD candidate at KIT in Germany. Today, I will present my findings on a cell death-inducing factor. I'm working with the unicellular green microalgae Chlorella vulgaris as model organism, and the cell disintegration method, the pulse electric field, short PEF treatment, is used. However, the biological processes involved are still poorly understood. Therefore, the aim of this study was to understand the biological aspects behind cell death after PEF treatment, and this might help to improve energy-efficient extraction from Chlorella vulgaris. Depending on the experiment, cultivation of Chlorella was conducted either under continuous light or with a light dark cycle, leading to synchronized culture. When cultivating under continuous light, the exponential growth phase after inoculation takes place until day 3, and the stationary growth phase starts on day 4. When applying a light dark cycle and continuous dilution, this leads to synchronized culture, where division of the cells is restricted to a short time period just before the onset of light. Samples taken from a specific time point always represent one developmental stage of the cell cycle. The culture is always in exponential growth phase and a steady state is never reached. To determine and monitor factors regulating cell death in response to PEF treatment, a robust method was established to measure cell viability after PEF treatment. This was based on the difluorescein D-acetate, which is a non-fluorescent and cell-permeating plant probe used for measuring plant viability. It's cleaved by intracellular esterases in living cells to fluorescein, which then can be measured using flow cytometry. PEF treatment can be applied at different specific energies. Right now we heard of very high specific energies. Very low energies, I was using 0.8 joule per milliliter in this case, only cause a small portion of cells to die. However, PEF treatment requires also high biomass concentrations to make the technology cost effective. And this set of experiment aimed at analyzing the influence factor of cell density on the treatment. When diluting high cell density suspensions directly after PEF treatment to low cell density suspensions, we could see a constant mortality of around 30%. However, at the high cell density suspension, we could observe a slow death over a time period of 24 hours. Since the cells had been separated only after pulsing, the physical parameters can be excluded as factors, and the cell death must arise from biological processes occurring after the pulse. This led to the hypothesis that a cell death inducing factor is released after PEF treatment. The experimental setup was designed to test the toxicity of an extracted compound in the supernatant, short SN, of donor cells on other untreated viable recipient cells. High cell density donor cells are PEF treated using low energies and then incubated for 24 hours in order to release this putative cell death inducing factor. This attained PEF supernatant is then used to resuspend fresh recipient cells, and then a viability assay is performed to see the survival. From that, the mortality is deduced from the viability, so that's why we always see the mortality on the schemes. In the first experiment, the varied parameter for our experiment was the specific energy used for PEF treatment. Donor cells were cultivated under continuous light and harvested 7 days post-inoculation. To resuspend in the PEF supernatant, recipient cells from synchronized cultures were used. And as negative control, the recipient cells were resuspended in their own medium or in the supernatant of cells without PEF treatment. As you can clearly see, the attained PEF supernatant from donor cells with the different energies clearly leads to cell death. So we can observe a strong and rapid mortality of the recipient cells in response to treatment with PEF supernatant. The applied specific energy has no influence on the cell death inducing effect, which is why we set a specific energy of 8 or 9 joule per milliliter for the subsequent experiments. In order to analyze the cell death inducing factor, we first took a look at the donor cells. The effect of a different cell disintegration method, HPH treatment, and the effect of heating of the PEF supernatant was investigated. Incubation of the PEF supernatant at 40 degrees and at 50 degrees for 2 hours prior to resuspension of the recipient cells almost completely disables the cell death inducing effect. Interestingly, the supernatant of the other cell disintegration method, high pressure homogenization, HPH, also leads to extraction of the cell death inducing factor. Therefore, we can conclude that the cell death inducing factor is extracted rather than generated in the donor cells, since the HPH treatment destroys the cell immediately and does not allow any cellular response to the treatment. The generation of the cell death inducing factor in the donor cells provides important information as well. For this purpose, donor cells were taken at different time points in the growth phase of the donor cell, and of course we normalized the biomass concentration. These results show that the cell growth phase of the donor cells plays an important role for the generation of the cell death inducing factor. The PEF supernatant only shows the cell death inducing effect when the donor cells are reaching stationary phase starting on day 4 post-inoculation. Our hypothesis concludes that the cell death inducing factor is generated due to stress or nutrient limitation in the donor cell. The reception of the signal in the recipient cells plays quite a big role as well. For that purpose, we further tested the dose-response relation of the cell death inducing compound by diluting the PEF extract from the donor cells while keeping the cell count of the recipient cells constant. The PEF extract from the donor cells retains comparable cell death inducing effect down to a dilution of 10 times. However, when diluting 50 times or 100 times, the effect starts to diminish. This dose dependency can be explained by a model involving a receptor that accepts the cell death inducing signal, but this receptor can saturate it with the signal at high concentrations. When diluting the extract by factor 50, this putative receptor does not seem to be saturated anymore, leading to reduced signaling and leading to reduced mortality. We wondered also whether the responsiveness of the recipient cells depends on the cell cycle. So for this purpose, the varied parameter was a time point in the synchronization cycle. As you can see here, this is the scheme showing the cell sampling. Generally, we can see that the sensitivity of the recipient cell was dependent on the cell cycle. Before onset of light at time point 0, the orange bar, the cells have a lower mortality rate compared to cells that are already a little bit larger, 6 hours after light induction, or even the cells that are shortly before division, minus 6 hours before light induction. Therefore, the responsiveness of the cell to the cell death inducing factor depends on the cell cycle stage. The high specificity and dependence on the cell cycle indicate a signal receptor complex again that either shows a different expression level or different activity depending on the cell cycle. Whether the concentration of receptor varies over the cell cycle or whether the transduction or the processing of the signal deployed by this receptor is cell cycle dependent remains to be elucidated. If the cell death after PEF treatment involves active biological processes rather than being a merely physical phenomenon, it might be sufficient to trigger these processes at low energy and see the same effect as for higher energy inputs. So for this experiment, we changed the experiment setup and used first a biomass of 5 milligram per milliliter. And if we stick to the labeling from before, now the donor cells and the recipient cells are in the same suspension after PEF. So we varied now the specific energy and determined the dose-response relation of protein recovery after PEF treatment. So we had a variance from 0.8 to 8 joule per milliliter, the energies, and then we measured both the mortality of the cells after PEF treatment as well as the protein content at 6 hours and 24 hours after PEF treatment. And this dose-response relation shows that the protein recovery was already reaching saturation with the second lowest energy that we tested, the 1.6 joule per milliliter. We can see a virtually identical protein recovery for these two samples. And the mortality was already quite high. So we don't need much energy to achieve the same protein extraction efficiency. The lowest tested energy shows the effect of the cell death inducing factor again, because we have the slow death. After 6 hours, only 24% are dead, and then after 24 hours, 54%. And after the 6 hour sample, you don't see any protein, but when the cell death inducing factor is at play and more cells are dying, we can also extract protein afterwards. And this is 2/3 of the protein of the total protein we can see in this experiment. So it's already quite good. This supports our hypothesis that the PEF treatment deploys active signaling, culminating in the observed induction of cell death in the recipient cells. That can be in one mixture and this low energy procedure can be beneficial for protein extraction. In summary, we were able to show that PEF treatment extracts a cell death inducing factor from Chlorella vulgaris in stationary phase. And the analysis from the donor cells shows that the cell death inducing factor is heat labile and the compound is extracted rather than generated. The analysis in the recipient cells showed that showed dose dependency as well as a responsiveness to the cell death inducing factor depending on the cell cycle stage. The high specificity of this process is consistent with a biochemical model where PEF treatment deploys active signaling, culminating in the observed induction of programmed cell death. Therefore, insight into biological signaling can help biotechnology to be more energy efficient and thus more sustainable. Thank you for your attention. Special thanks go to my group for supporting and advising me. And if you have any questions, please feel free to ask. Thank you.
Thank you, Damaris, for this very interesting presentation on these applications of PEF in the case of microalgae to induce this kind of stress, which that lead to the cell death. I don't know if there are questions in the, probably there is some questions in the chat. From Stefania Romeo. I don't know if you want to read by yourself.
Yeah, I'm just trying to see the questions that regard me. Similar effect called bystander effect has been observed also with other types of signals like ionizing radiation, RF radiation, and PEF as well. Can you discuss? I didn't know that it has been observed for PEF as well. So I've already read some literature regarding that Chlorella vulgaris in stationary phase, that they that there's a substance that was later termed chlorelline that inhibits the growth of Chlorella itself. So this could be connected to that as well, but the bystander effect, I will actually look into that. Thank you for the suggestion. And the PEF treatment didn't extract cell death inducing factor, but induced the secretion of this factor. So that's the different wording, extract, but induce the secretion. So that is what I tried to show with the HPH experiment. So with the PEF treatment, of course, we can trigger signaling, especially if we are working with very low energies. But the PEF treatment that we used was leading to immediate cell death normally. And therefore, that's why we said we extracted. But with the HPH treatment, definitely no cellular response can be seen from the cell. So we see the effect as well, and that's why we said it's extraction, but not in. I don't think we can say induction then in that case, because also we can see the effect for the HPH supernatant.
If you can please answer to the last questions, because you were a little bit late, from Javier Raso.
Yeah. Of course, yes, I have tried to identify the molecule, but so far I haven't had success yet. So I did some size chromatography because first we went into the direction of protein, but there was no clear result. So right now we're looking into volatile molecules actually, because there's also the smell after PEFing the algae. So we're looking for some gas chromatography results, and this is in progress. But so far we don't have any clear candidate yet, unfortunately, but we're also very interested in that. Thank you for the question.
Thank you again, Damaris. Okay, we can now move to the last speaker of this first part of dedicated to food technology. She's Dr. Sumio Kanafusa. She got her PhD in agricultural science at the University of Tsukuba in Japan, and now is currently visiting researcher fellow at the Department of Food Technology, Engineering and Nutrition of Lund University. And she will talk again, continuing on the inducing stress response on microorganisms, in this case on lactic acid bacteria. She will present a work titled The Effect of Nanoseconds Pulsed Electric Field on the productions of metabolites from lactic acid bacteria. So, Sumio, you can switch on, please, the microphone and camera.
Hi,
Thank you very much for
inviting
giving me such a wonderful opportunity.
You can start, please.
Today, I'd like to talk about the effect of nanosecond pulse electric field on the production of metabolites from lactic acid bacteria. First, I'd like to talk about basic fermentation profile of Lactobacillus plantarum in watermelon juice. After that, I'd like to share with you some of the interesting data of nanosecond pulse electric field treatment in fermented watermelon juice. Watermelon juice is a healthy drink rich in antioxidant and bioactive compounds. However, it is sensitive to heat, oxygen, and light during food processing and generate unpleasant flavors. This research focused on lactic acid fermentation as one of the food processing for watermelon juice. The fermentation process improved preservation of watermelon juice and also improved nutritional value by producing metabolites. The metabolites produce special flavor, and fermented food enhanced nutritional properties. Lactobacillus plantarum is one of the common anaerobic bacterium for lactic acid fermentation. It has been studied for its potential as probiotics. L. plantarum produced lactic acid, acetic acid, and ethanol as some of the metabolites. It has been reported metabolites of L. plantarum has healthy promoting effect. Improving fermentation efficiency has been studied. Gene recombination increased production of lactic acid in yeast, and nanosecond pulse electric field increased ivermectin in streptomyces. In our study, we applied non-lethal nanopef on L. plantarum in watermelon juice. The objective of this study is to evaluate nanopef effect on the production of fermentation metabolites and cell viability. Before PEF treatment, we measured basic fermentation profile of L. plantarum in watermelon juice. Watermelon was crushed and vacuum packed in plastic bag. The juice was sterilized and immediately cooled before inoculation. L. plantarum was inoculated to sterilized watermelon juice. Then the juice was incubated at 30 degrees for 20 hours. After inoculation, growth, pH, and concentration of lactic acid and acetic acid were measured. This figure shows cell growth and pH of fermented watermelon juice without PEF treatment. The process of fermentation is shown by an increase in cell count and decrease in pH. Cells reach the stationary phase after 18 hours. pH was decreased dynamically and reached to 3.8 after 20 hours. This result indicated that lactic acid fermentation proceeded in the watermelon juice. Production of metabolites from L-plantanum without pH treatment are shown in this figure. The L-lactic acid increased during the log phase. Acetic acid started to increase 16 hours after inoculation and was found in a small amount. This result shows that typical metabolites of lactic acid bacteria increased during the log phase in watermelon juice. From this result, we decided nanopEF treatment was performed at the log phase of 15 hours after the inoculation, when the pH changed drastically. Next step, we applied nanopEF treatment to fermented watermelon juice. The watermelon juice was placed in cuvette and incubated at 30 degrees for 15 hours and treated with nanopEF. The operating electric field condition was as described here. After PEF treatment, the juice was incubated at 30 degrees for 5 hours. 3 repetitions were performed for each PEF protocol. The fermented juice was used for growth and lactic acid and acetic acid measurement. First, we measure the frequency effect. When the frequency increased from 1 to 50 Hz, the concentration of D-lactic acid and acetic acid significantly increased compared with 1 Hz. These results suggest that the increase in metabolite was caused by the increased current flow to the cells. From these results, we decided nanopEF treatment was performed with 50 Hz. What you see here is effect of voltage and the total number of pulses on D-lactic acid production. The horizontal lines show the production by PEF untreated samples. When the frequency and number of pulses were kept constant, but the applied voltage was 4.5 kilovolt, the concentration of D-lactic acid was higher than the untreated control. When the voltage was fixed at 4.5 kilovolt, increased the number of pulses to 700 provoked 6.8% increase in D-lactic acid. Similarly, regarding L-lactic acid production, when the voltage reached 5 kV, L-lactic acid was increased, but the voltage was decreased to 4 kV. 12% decrease was observed. When the voltage was fixed at 5 kV, increased number of pulses to 700 provoked 19 increase in L-lactic acid. Regarding acetic acid production, the concentration of acetic acid was significantly higher than the control when the applied voltage were 4 to 6 kV. When the voltage was fixed at 4.5 kV, 11 to 15% significant increase was observed. Acetic acid increased regardless of voltage and number of pulses. Regarding cell growth, changing the applied voltage and number of pulses did not result in an increase or decrease cell count compared with the untreated control. So, nano-PEF treatment did not affect the cell growth. Here is a hypothesis of the result from nano-PEF stress. It has been reported that loss are produced after PEF treatment and caused the direction of metabolic fluxes. In this case, increased production of metabolites was not accompanied by increased cell growth. And it is also has been reported that metabolism pathway changes in stressed cell of lactic acid bacteria. This research showed nano-PEF treatment increased lactic and acetic acid in watermelon juice without affecting cell growth, and suggested that metabolic fluxes of the bacteria were redirected by nano-PEF. Further studies are needed to better understanding the mechanisms. This knowledge would aid future research for producing high-quality fermented food. Our research article is published. This work was collaborated with Lund University and the National Food Research Institute in Japan. I appreciate all the persons for helping our research. Thank you very much for listening to my presentation.
Thank you, Suimo, for this very nice presentation. We are very late, but if there is one, maximum two quick questions, I cannot see in the chat. I have just one question. Just do you try also to use microseconds instead of nanoseconds? And why did you choose to use nanoseconds?
Yeah, I try to use the microsecond, but I really want to see to use new technology. So that's why I want to use nanosecond PEP. Then, yeah, it's a very good suggestion. Then I'd like to compare the micro second and the nanosecond as well. Thank you.
Right, thank you very much. Thank you to everyone. We have a 10 minutes break according to the program, and we'll see again here at 3:25, just 10 minutes break. Thank you.
You may start now.
Hello, everybody. Again, we are going to start with the second part of the webinar that is devoted to basic science. Before introducing the first speaker, I would like to remind you that in order to prevent technical problems, it has been decided to switch off the microphones and the camera of attendants. So please use the question and answer tools for asking questions to the speakers at the end of their presentation. Okay, so I'm going to introduce the first speaker. It's a big pleasure for me to introduce all of you to my colleague, Antoni Iborra. Tony is full professor at the Department of Information and Communication Technologies of the University Pompeu Fabra in Spain. Tony's research is focused on bioelectrical phenomena with the purpose of implementing new methods and devices for biomedical application. And he is inventor of a number of patent applications. And I think it's remarkable that he was awarded in 2016 with a grant from the prestigious program of the European Union IRC Consolidator Grant. Tony, the ground is yours.
Thank you very much for the presentation, for the introduction. Thank you very much for the opportunity. I'm going to share the presentation. So hopefully you see it. So it's an honor to be here. It's a pleasure. And thank you very much. So hello, everybody. Good morning, good afternoon, good evening. So what I'm going to talk here today, it's in part was presented in the last EBTT course in Ljubljana. So apologies for those that were there, because this is basically going to be a repetition of what I presented there up to a point. So, and the talk is about avoiding nerve impulses so meaning during the delivery of the pulses for electroporation, and avoiding nerve impulses by electroporation. So it's going to have two parts. But before, just a brief detour, that I'm going to, the talk is about avoiding nerve impulses, but most of my recent work is about actually inducing nerve impulses. And this is related to the project, the ERC project that Javier mentioned. And what we are doing in this project basically is developing microstimulators that are going to look like what you see here, thread-like stimulators. So they are going to be injectable. And these stimulators are going to be used for neuroprosthetics. The idea is that we want to perform what is called functional neuromuscular stimulation for recovering movement in patients with paralysis. And the idea here, new thing, is that instead of using the standard or most common ways of powering the electronic implants that are batteries, inductive coupling, or ultrasound transmission power, what we are doing is to apply high frequency currents through the tissues. And these high frequency currents, because they are high frequency and we apply them at short episodes and short bursts, they are innocuous, you don't even feel them. And then what the implants do is to transform these high frequency currents by rectification, by electronic rectification. They are transformed into low frequency currents, which are capable of performing local stimulation at the place where they are implanted. The nice thing here is that because we don't have any batteries, coils for inductive coupling, piezoelectric crystals for getting the power by ultrasound, we can make these implants very thin because this is purely electronics. And this is where we are working. So this is one of the last things that we have done, just an update for those of you that are more or less familiar with the project and maybe are curious about it. So we are in the right, let's say, direction. So we already produce prototypes which are very similar to those that would be the clinical prototypes at some point, future years, I hope. And basically what we have is a capsule, a titanium capsule that contains our electronics. So we had to develop our own integrated circuit for that. And then we have two electrodes. All those materials are materials and technologies, actually, the fabrication technologies, let's say, that are common in implants. And what we do is to inject them. We did a test last July in which we actually injected them in a rabbit leg, a number of them. And then we were able to power and communicate with them. So we were able to activate one implant or the other to perform the stimulation of one muscle or the other, just by computer. So after that, I come back to the topic of my presentation, which has two parts. As I said, first I'm going to talk about nerve impulses in electroporation and then by electroporation. So just a recap of the two phenomena. Electroporation, it's well known for the audience here. And electrical stimulation, I guess, is also well known for most people here. Electrical stimulation means basically applying electric currents to artificially and in the body to artificially induce action potentials. So action potentials are self-sustained perturbations of the transmembrane voltage that travel through the membrane. So that's what it's a nerve impulse. A nerve impulse is a perturbation of the transmembrane voltage, a local perturbation of the transmembrane voltage that is able to propagate. And this is the basis of the communications through the nervous system. The nervous system basically is made of neurons. Neurons have long projections that are axons, and that's where the, let's say, signals travel. This action potential perturbation, propagation or generation actually happens only in specific cells. It does not happen in all cells. It's not as universal, let's say, as electroporation. It happens in neuron and in muscle cells, including myocytes. There is a very important common point with electroporation, and it's relevant for the rest of the talk, which is that electrical stimulation also happens when we perturbate the transmembrane voltage by the delivery of this current, external current, which generates an electric field. So it occurs when we perturbate the transmembrane voltage beyond the threshold. So once we perturbate the transmembrane voltage beyond the threshold is when we have an action potential triggering and then the propagation. There is typically, or not typically, actually, there is an important difference, which is that the threshold for inducing electroporation, so this perturbation of the transmembrane voltage that we have to induce to cause electroporation, it's substantially higher than the threshold that we need for electrical stimulation. Here, we are talking about an excursion between 200 millivolts and 1 volt, depending on how you assess electroporation. Here, we would be talking in a range of 50 millivolts, 100 millivolts most top. So that means that it's very difficult to do electroporation without inducing nerve impulses. So basically, when we apply conventional pulses for electroporation, meaning 100 microsecond pulses, 10 microsecond pulses, monophasic pulses, we basically very easily are going to activate nerves. So the nerves are bundles of axons. So very easily we are going to activate nearby nerves very easily. And this is going to cause pain and muscle contractions. And that's why anesthesia and muscle relaxants are used in a lot of clinical applications of electroporation. In the past, there were ideas to minimize the impact of those contractions, at least the part of the contractions. The pain basically has only been solved with anesthesia. The muscle contractions, the ideas that were there were basically to, instead of applying the pulses at very low frequency, which could cause multiple contractions, to apply them very fast. So we would have a single contraction. The problem with that probably is that we have a larger contraction. And then the other idea was also to apply stimulation before the pulses for electroporation, so that the muscle was fatigued or it was already contracted, so that when nerve impulses reach the muscle, were not causing so much movement. This works, but this is actually not avoiding the nerve impulses. What was avoiding the nerve impulses, or at least minimizing their impact, was a protocol or a waveform proposed and developed by the group in Virginia Tech, by the group of Rafael Dablos, like 10 years ago, with the name It's Fire, which basically consists in high frequency biphasic pulses, square biphasic pulses. And this, this comes from the fact that high frequency signals, high frequency currents, typically are not able to stimulate, or are less susceptible to stimulate. That's what we are using in our microstimulators technology. So at first glance, it sounds reasonable, but at second glance, let's say it's not so reasonable, or not so easy to understand that it will work, because both phenomena, electrical stimulation and electroporation, are dependent on the transmembrane voltage. So we increase, let's say we can increase the current for stimulation by increasing the frequency, because we basically are reducing the transmembrane voltage. There is a low pass filter phenomenon. So the same should also be expected for electroporation. So as I said, at second glance, it's not so obvious that these high frequency currents or high frequency square wave could do electroporation without inducing nerve stimulation or reducing a lot the level of electrical stimulation. So that's, I was puzzled, and I basically puzzled my team, let's say, to work on that. And also we started a collaboration with Virginia Tech, with Professor Davalos on that, to try to understand what's going on there. And what we did, what Borja Malcanal actually did, it was to create a model of tissue in which there are nerve terminations and there are axons, also there are nerves. And then we had 2 electrodes to apply our voltages for electroporation. So, and we tried different waveforms. How we did that? So basically, what we did in order to compute or predict electroporation, we followed the standard process, which is basically to simulate the electric field distribution. And then we computed the areas that were electroporated as the areas where the electric field was beyond a threshold for one specific protocol. So these thresholds were obtained from experiments, previous experiments. Then what we added, what is new, is that we added a cable model, what is called a cable model, which is a model for axons, and also it serves also for nerve terminations. And this model, I'm not going to go into details. Basically, what it models are the nodes of Rambier, and how these nodes of Rambier can trigger, because the channels they can trigger an action potential, and this action potential can travel. Actually, this model allows us to see the evolution of the action potential and how it propagates. The only input, let's say, are these external voltages, which are the external voltages caused by the electric field distribution. And so we started to study what we obtained. The first thing that we obtained was that what we expected, that if we use monophasic pulses, so these 2 points here, this is the pulse length, this is the voltage that we need for stimulation. If we use monophasic pulses, we very easily are going to activate nearby nerves. Those are nerves or axons, better said, at 1 centimeter. And just with less than 10 volts, we activate it. So there is, let's say, no chance of electroporation without inducing a stimulation. But as we increase the frequency, well, for biphasic pulses, we see that we have to increase more and more we can increase more and more the voltage that we apply between across those 2 electrodes before inducing a stimulation. If now we use those thresholds for stimulation and use them to compute the ablated area according to the empirical thresholds, what we see is that when we reduce the pulse length, there is a point in which we can start to obtain significant volumes ablated. So we can ablate, let's say, almost 1 cubic centimeter without inducing a stimulation of these 2 nearby nerves. Another thing that we did was to do the same for nerve terminations. Nerve terminations are a little bit more interesting, at least for illustration, because for axons, the threshold, it's a threshold for stimulations, a threshold that depends on the derivative, the spatial derivative of the electric field. On the other hand, the threshold that we have for nerve termination is also an electric field magnitude threshold. So it's comparable to the threshold that we have for electroporation. So we can compare them at the same in same terms, let's say. And that's what we did. What we did was for a 2000 volts pulse, monophasic pulse, we computed the area that we would be electroporating. So this is the area that we would be electroporating or damaging or destroying by IRE. And then we also computed the area in which we would have stimulation of any nerve termination around here. So it's a huge area. So meaning that it's very easy to stimulate nerve terminations. On the other hand, if we go to these H5 protocols, in particular this one, for instance, if we want to treat the same area, we have to increase the voltage. So now we have to go from 2000 volts to 9500 volts. But what we see is that the stimulation area, the area where we are going to stimulate nerve terminations, is much smaller. So again, we see this phenomenon, which it shows that we can achieve electroporation without inducing or minimizing the chances of stimulation. And why is that? Well, the reason for that is that if we look at this data for nerve terminations, is that what we see is that the threshold for stimulation increases with frequency. So this is the inverse of the pulse length, increases with frequency as we would expect for low pass phenomenon. On the other hand, the thresholds for IRE also increase with frequency as we expected, but they increase with a lower pace for frequency. So there is this increase, it's less step, let's say. And this is the reason. So basically, we can get to the point of working at very high frequencies, so very short pulses, so that we can still induce significant electroporation, inducing or minimizing the chance of stimulation. And well, why there is difference in thresholds? As I said, that's not obvious. We have an explanation, which actually we have not proved, and this is kind of an hypothesis. Our hypothesis is that this is related with the different charging time of the axons compared to the cells that we treat. So the cells that we are going to treat probably are going to be more or less regular in size. All dimensions are going to be, so we can simplify them as round small cells. Whereas the cells that we are stimulating, the axons, are long cells. So long cells are big cells, and big cells, if you recall Sean's equation, basically are supposed to be easier to electroporate in the sense that the transmembrane voltage that we can reach in a long cell or a large cell is going to be larger than the transmembrane voltage that we will reach in a small cell. So, and that's what we see here. If we look at that simulation, we see that the transmembrane voltage that we have in this long cell, it's larger than the transmembrane voltage that we have in that small cell. But the thing here is that that's true for long pulses. Okay, this is a 100 microsecond pulses. If we look what happens at the beginning, what we see that what happens at the beginning is that the small cell, the small round cell, charges very rapidly compared to the long cell. So basically, if we are applying very short pulses, we would reach, let's say, thresholds here for stimulate for electroporation here in the round cells before reaching thresholds, let's say, for stimulation in the long cell. That's what we believe. Another way of saying that, it's applying biphasic pulses. So now we are looking at biphasic pulses, the same model, just looking at the transmembrane voltage, no electroporation seen here. And what we see is that if we look at the transmembrane voltage in the small cell, we see that as we increase the frequency, so as we reduce the pulse length, more or less the magnitude is constant. Whereas in the case of the long cell, we see that there is a dramatic drop in the magnitude. So basically, this is consistent with this behavior that we saw before, in which the thresholds for stimulation rise, let's say, very rapidly with frequency, whereas the thresholds for IRE basically are flat or increase slowly with frequency. Okay, so that was the first study. I'm going to present kind of rapidly 4 studies. The second study, actually, I'm going to go super fast. And this is just to illustrate that after noticing that biphasic square waveforms can be used for stimulation and for electroporation, reducing the chance of stimulation, we wonder whether sinusoidal waveforms could do the same thing, because actually, according to simulations with the cable model, they should have a similar behavior. And this is basically the work of Quincas TV. Very rapidly here, what we did was to demonstrate that we can do electroporation in a rabbit liver, minimizing the chance of stimulation. Very similar with sinusoidal waveforms. Sinusoidal waveforms may have some advantages compared to square waveforms in terms of generation and electromagnetic compatibility. I don't want to go a little too much into details. So basically to say that in this particular setup, beyond 50 kilohertz, we basically didn't see any contractions anymore. And at the same time, we were able to electroporate. So that's another way. So work with sinusoidal waveforms, sorry, instead of square waveforms. Okay. So that, let's say, concludes the first part of my talk, which was about minimizing or avoiding stimulation during electroporation. So now the second part is about avoiding nerve impulses by electroporation. So this is quite obvious. I mean, for anybody that has intellectual work or study on nerve physiology, to know that if we increase the conductivity of the membrane, if we make it more permeable, what we are going to see is that the nerve impulses or the action potentials don't propagate or reduce their speed. Actually, there is going to be a drop in the speed, and then there is going to be a complete blockage of action potential propagation. And this is something that the cable model predicts, and as I will show you in 2 slides. What you see here, this is, let's say, a schematic representation of an axon with myelin, so a myelinated axon, in which the action potential basically travels from one node of Rambier, these are the nodes of Rambier, to the next node of Rambier very rapidly. There are other fibers in which there is no myelin, and the propagation is continuous and not slower, basically the C fibers in the nerves. So that's, as I said, that it's something expected, that's something known, and actually was demonstrated in the 90s. For instance, the group of Rafael Lee in the 90s demonstrated that applying pulses for electroporation, it's possible to block the conduction in nerves. And this is, in some cases, a reversible phenomenon in which after a few minutes, hours, it's the normal velocity, it's recovered. Nice. We were a little bit more than studying this fundamental blockage in electroporation, which is also interesting to see what happens with the nerves and the preservation of the nerves when we apply electroporation, because we know that we can apply electroporation and the nerve function is going to be there, to be gone for hours, even days, and later recover. This is probably related to regeneration because of the fact that we don't damage the matrix with reversible electroporation. That's a very interesting topic. But we were more interested, I was more interested in studying the possibility that electroporation is involved in other bioelectric phenomena. Okay, in particular, bioelectric phenomena related to pain or therapies, let's say. One of these occurrences of nerve block that we can induce artificially is what it's called high-frequency alternating current stimulation. This is basically the KHFAC stimulation. We're talking about a frequency in the order of 1 to 10 kilohertz. This is the work of Sergi Marcoval and Tomás Garcia. This is a very preliminary work that we did just very recently to see if there was any potential to electroporation to be involved in that, any potential for direct electroporation, let's say, to be involved in that. Because the setup that you see that researchers in this area demonstrating that they can block both afferent and efferent nerves, meaning nerves that go to the muscle and nerves that receive sensory information. So that has been demonstrated. And researchers working in this area, they develop setups like this one, in which you have a nerve, a stimulator here, proximal stimulator, an area where you perform this high frequency stimulation, and an area where there is a distal conventional stimulator. And when you apply impulses at, let's say, 10 hertz from the proximal stimulator, this reach the muscle, this is muscle, that stimulates the muscle, and this basically causes a force, and they see the peak of forces. Once they activate this high frequency waveform, which is moderate in magnitude, very moderate in magnitude, they see that the nerve, there is no muscle contractions. And this muscle contraction is not because we damage the muscle or we damage the neurotransmitter or something like that, because actually if you stimulate here, the muscles contract. And after removing this stimulation, the blockage is reversible. So showing that the nerve blockage is occurring here very, very clearly. So to us, it was more or less evident that electroporation could be involved in that. And we are a little bit surprised not to have identified any paper suggesting that. Though I'm still very skeptical that we didn't find any paper saying that. And basically, we wanted to see if electroporation could be involved. And we did a very simple study. Again, we took the cable model. And what we did is in the cable model, play with the conductivity of a segment. Okay, a number of, let's say, nodes of Rambier. And we observed, so the conductivity of the membrane. And then we studied the velocity. We applied stimulation pulse and action potential, and we saw how the action potential traveled. And we saw that, as we expected, the velocity drops when we increase the conductivity of the membrane, of that segment of the membrane. And there is a point in which actually it's blocked. There is no transmission of the action potential anymore. And then what we did it was to apply a model for electroporation of the axon. This is the model of Gregorin and Grasowska with the same cable model. And what we did was to apply voltages that are, let's say, protocols that are similar to this one or equivalent to those ones that are used in this high frequency AC stimulation. And what we saw is that we basically reached membrane conductivity values that are very similar or even go beyond that conductivity that we obtained before for blocking the action potential propagation. So, as I said, this is very preliminary, but it points to this, or it seems to indicate that probably electroporation has a direct impact on the membrane conductivity, and that's what is causing the mechanism of action of this KHF-AC stimulation. So another thing that actually we did before that, it was to study the possible role of electroporation in a therapy, which is called PRF, for pulse radio frequency, which is a technique that applying quite high frequency with a bipolar needle, it's able to treat pain. Yeah? Principally, most of the times it's applied close to the spinal cord or the ganglia, but in some cases it's also applied in other spots of the peripheral nerves. And this has an effect, a beneficial effect in terms of pain that can last for months. So the fact that it's lasting for months indicates that it cannot be direct effect of electroporation. The increase in permeability, it's going to be minutes, hours at most. So we don't expect that the electroporation can have a direct effect on that. But we still thought that maybe there was an indirect effect. And the reason for that is the following. So this, I'm summarizing what we know about PRF, what it's known. Again, this is basically the work of Borja. What we did, what we know is that this is the protocols that are applied. We are talking about 50 volts typically. So that's for the geometry of the needles. These are fields that are around 100 volts centimeter to 100 volts centimeter. So at high frequency, no suspicion at first glance of electroporation. But still, so what is known is that it works. So that's work, and it's quite clear that it's not a placebo effect at this point, because with pain we always have to be careful with that. So it lasts for months, as I said. The mechanism of action is actually not understood. There are some observations that I will just point below. And what is also known is that it has to be an effect on C fibers because of the kind of pain that it's relieved. This is the dual pain being related to C fibers, which are non-myelinated fibers. As I said, what is known is that there is no permanent damage. It's not thermal damage. It's affecting C fibers. And there are different observations at the level of the cells that made us thought that could be related somehow to calcium. So, and what we hypothesize, basically not to speculate, what we think is that we have calcium getting into the cells by something that we refer to mild electroporation, because actually, as I said, the electric field is very low and we are at high frequency. And with this hypothesis, we try to understand if that makes sense. So we did an experimental study to see if there was any rationale behind this hypothesis, or it was somehow plausible.
Tony, please, while your time is almost over, can you finish in a couple of minutes, please?
Yeah, I'm going to try to rush as much as possible.
Okay.
So what we did, it was in an in vitro study. We basically applied YoPro and calcium indicator. So basically to see if calcium was getting into the cell. So I'm going to go very rapidly. And then with that, what we saw is that we have just a logistic regression. And then we were able to see that calcium was getting into the cell. And this hypothesis of the mild electroporation made sense in the sense that YoPro was only getting to very high electric fields, whereas calcium was getting for low electric fields. Okay. Then we also saw that this calcium was getting from the outside, not from the outside. It was not a relief from the plasmatic reticulum. And another thing that we studied, it was that it was not thermal, because as I said, it was not thermal at the level of bulk temperature, but it was hypothesized that it could be related to spikes in temperature, which indeed occur very close to the needle. So this we also tested with a dye that we have spikes in temperature, and it seems like it could be the case that these spikes in temperature had some effect, and those spikes in temperature could be causing the increase in calcium. So basically, what we did, it was to confirm that temperature had nothing to do, because if we applied, when we applied in a different assay, different frequencies, we saw that different frequencies were inducing, were having different responses, whereas if it was thermal, the effect would be the same. At the same time, we studied at 2 frequencies and 2 conductivities, and these differences that we obtained here are completely compatible with the differences that we would obtain for a phenomena that is related to the transmembrane voltage. If we see the transmembrane voltage, so we simulated the transmembrane voltage for 2 conductivities and different frequencies, what we see is that at low frequencies, basically there is no differences as we obtain experimentally, whereas at high frequencies, we see that there is a difference for the 2 conductivities. So this indicates that this is a transmembrane related phenomenon. So basically, we are very convinced now that PRF bars cause an uptake that of calcium that appears to be related to electroporation, something that we refer to as mild electroporation. The question is, or one question is, why it's only affecting C fibers? Well, there are different explanations, and not alpha, delta, and alpha, beta fibers. So different explanations. One could be that the C fibers are not protected by the myelin, so there's more chance that calcium gets inside. Another is related to the thing that I discussed at the beginning about H-fire, in the sense that long cell, the nosotropin has a high resistance, so they are slower to charge, and there could be other physiological reasons. So to end, let's say a take-home message is that maybe the role of indirect or direct of electroporation in electrical stimulation therapies and in bioelectrical phenomena in general has been overlooked. If we look at these electrical stimulation therapies, what is defined in the literature, in the classification mesh definition. So we have discussed here about pulse radio frequency. Transcutaneous nerve electrical stimulation typically is done at low frequencies, not with these high frequencies, but in some cases also. And here we partially indicate that that may be related, the electroporation may be related. For the case of electric puncture shock, this is defibrillation. The role of electroporation was proposed long ago, and there are strong evidences that it can be indeed related. But what happens with deep brain stimulation is we still don't understand the mechanism of deep brain stimulation, and maybe it's related. The thresholds, at the end I said, the thresholds for electrical stimulation are lower than the thresholds for electroporation, but are not so different. And we see here that we can still, with very low electric fields, with this mild electroporation, we may be doing something. Then maybe there is a role in spinal cord stimulation for pain relief. We don't know. There may be a role in vagus nerve stimulation. With that, I conclude. Just thank my team and all the past members and present members and the agencies that have funded the research that I presented here. And thanks to you, and apologies for the delay. Sorry.
Okay. Thank you, Tony, for your presentation. Very interesting presentation on a topic completely different of the former presentation. So that demonstrates that electroporation is a cross-disciplinary topic. So there is a couple of questions in the section question and answer. So can you see the questions?
Yeah,
can you answer?
Yeah, the first question was, what about electrical stimulation of cardiac cells during cardiac electroporation? Well, yeah, we can have stimulation of cardiac cells. It's very important to synchronize where you are applying the pulses, so that you don't cause new action potentials through the cell. So if in the cardiac cell, there's going to be a period in which is called the absolute refractory period, in which if you apply pulses, you are not going to cause any other action potential. So it's a safe, let's say, window. Okay, so that's the first question. Can the electric pulse that block nerve conduction or reduce the speed of conduction be used to treat it? Well, I hope this is more or less now answered in the presentation. Yes, we believe that they may be related. Could you please share more details about the temperature tie? Yeah, I will share that through the chat. And then Yeah, exactly. Electroporation was suggested. Damian is pointing out to the fact that, yeah, electroporation was suggested as a mechanism for cardiac defibrillation. And yes, that's known. As I said, there are strong evidences that that's the case. So I think there are no more questions. Javier? There's another question. Does pulsing in the PRF play an important role in the calcium uptake and other things? Could continuous wave achieve the same response? Well, continuous wave could maybe achieve the same results according to what we see, but the problem is that continuous wave for sure would cause thermal damage. Okay, and that's actually, they reached let's say the researchers that got to the point of developing this PRF, before they were using continuous wave. And they were using continuous to cause damage. And at some point, somebody had the idea of making this burst, thinking that they would not induce so much damage. And then they discovered that they were not doing any thermal damage anymore, but still they had some effect. And that's when everything was not clear at all what was going on.
Okay, thank you very much, Tony. I had a problem with the connection, so I think we can finish your presentation. I suppose that if someone has some question, they can do the question to you by email and so on. Yeah, so we are going to pass to the next presentation. The next speaker is Siki Wow. He is a research associate professor at Frank Ridey Research Center for Bioelectrics in Old Dominion University in USA. And his research is focused in nanosecond electric pulses, tumor ablation, gene electrotransfer technology, and the induced immune response. So I think that the presentation is recorded in this case. Okay. No.
Hello, everybody. First thing.
Yes.
Hello, everybody. First, thank you for Javier's introduction. So then let me share screen first. Okay, so then I'm going to talk about nanosecond electrical pulse and its impact on the tumor microenvironment. As we know, tumor microenvironment is a major hurdle for the immunotherapy. And because the tumor microenvironment form called immunosuppressive network, those networks including immunosuppressive cells, cytokine, chemokine, exosome, and extracellular matrix. You can show here the major immunosuppressive cells are shown, such like regulatory T cells and myeloid-derived suppressor cells and the tumor-associated macrophage. And in some cases, such like pancreatic cancer-associated fibroblasts and tumor-associated neutrophils also participate. So we know for different type of cancer, their report have different immunogenicity. As shown here, usually we know that melanoma and colorectal cancer called hot tumor because they have a higher mutation burden and have a relatively good response to the immunotherapy. And then if you look at the pancreatic cancer and the breast cancer, they have very few mutation burden and also poor response to the immunotherapy. So we know previously we report nanosecond electrical pulse can induce immunoprotection in two poorly immunogenic cancers. One is 41, another PAN02. Both are have few mutation and resistant to immunotherapy. So as you can see here, for the 41 model, you can see here after primary ablation, then the second challenge, nanosecond treatment actually protect more than 93% of the animal And in case of the pancreatic cancer, nanosecond electropole treat 100 nanoseconds can protect 50% of animal. And however, the 200 nanosecond and IRE did not show any protection. So we go further, look at the difference between the systemic and the local change of the immunosuppressor cells. As you can see here, for both, we have a 41 condition have induced immune protection, and the panel 2 with parameter do not induce immunoprotection. So systemically in the blood, you can see the both model, two type of immunosuppressor, regulatory T cell and myeloid suppressor, they go significantly decrease, they both significantly decreased. And so that probably is the tumor burden effect. And so if you look closely, the change in the tumor, they show difference. In the protection model, both suppressor cells significantly go down after 1 day. But if you look at the non-protection, in the tumor, the suppressor regular T cell actually go up at the day 2, and the myeloid deployed suppressor drop a little bit, but not as much as protective 41 model. So that indicated the tumor microenvironment may be the reason causes a difference of two models. To look at detail for the tumor microenvironment, we have simple design. You can see here, first we give the tumor initiate, then treat with another second electropulse, then we look very earlier until 1 week, 4 hours, 8 hours, 1 day, 3 days, and 7 days. Have those tissues, look at the change in the local and systemically. Major focus on the tumor microenvironment. So this figure shows the overall change of the nanosecond electropulse treatment. You can see here the most significant actually is regulatory T cells, and the CD4 T cells, they dramatically decreased even after 4 hours. So, very significant. And also pay attention to those the ratio. You can see at the beginning, the regular T cells, roughly 50% of CD4 cells. So it indicated they are very immunosuppressive. Then after treatment, both recover a little bit, but you can see here have more CD4 cells than regular T cells. Like at the beginning, 50%, at the end, day 3 or 1 day, probably regular T cells only 20% of the total CD4 cells. Then if we look at the other two suppressor cells, they have also showed slightly different dynamic change. For the tumor-associated macrophage, after 1 day have significant drop, then maintain. But for the myeloid deployed suppressor, they only after 3 days showed a significant drop. Then surprisingly, if you look at the draining lymph nodes, the dynamic is different. If you look at the lymphocytes, CD4 cells actually significantly go up after treatment. But then if you look at regular T cells and the CD8 T cells, there are not much difference. So we suspect the significant drop of those suppressor cells probably killed by nanosecond electropulse. Then next we look at those death markers. So interesting, if you look at regular, first we look at Tregs. They actually did not show the death marker of Nexin-5. You can see here, they actually slightly decrease. So we don't know, because we know that cells can be killed, but it might be too earlier. Cell die too early, we cannot catch it, or they might migrate to the other organs, such as lymph nodes. Then we did see the tumor-associated macrophages after even 4 hours, majority of cells showed cell death marker. So, and the second electropulse definitely kill macrophage, but then may or may not regular T cells. Then it's interesting, we look at the cell death marker in the draining lymph node. First, we look at the regular T cells, actually showed the significant increase in the cell death marker. And most, after 1 day, the most regular T cells showed earlier cell death marker. And in contrast, for the tumor-associated macrophage, most of the cells showed later, both later and earlier evaporated cell mass was showed. And also, interestingly, in the draining lymph nodes, if you look at CD8 and all conventional CD4, it means CD4 positive, CD25 negative. they actually almost no effect. Look like regulatory T cells is the one die, and also especially CD44 or positive or active regulatory T cells are most vulnerable. Then if we look at the tumor-associated microarray, very interesting, if you get those live cells, the co-stimulatory molecules, MHC2 and CD86, goes significantly upregulated. So indicate tumor-associated macrophage polarized from M2 to M1, or for immunostimulation type. So as a result of this differential cell death, because look like nanosecond selectively kill regular T cells. So the result is actually CD4 or convention affect CD4 T cells relatively go up. So the ratio is also significantly increased. So it means good for the immunostimulation. Then the same thing pattern also occur in regional lymph nodes or draining lymph nodes. You can see here regular T cell significant drop, and also the ratio of the convention CD4 versus regular T cell significant go up. And the same thing in the systemic, in the blood, the regular T cell significant go down, the ratio of the convention CD4 and regular T cell. So those also show as a good sign for over times of immunosuppression. Then we know the function of a regular T cell is very important. It is the most potent to suppress cytotoxic T cells. So we perform the regular T cells function assay, as you can see here. If no regular T cells, cytotoxic CD8 T cell will have a huge proliferation and division. Then if you co-culture with a tumor source of regular T cells, most of the cells actually suppress, cannot proliferate. However, if you compare to the nanosecond pulse, same cells recover a little bit. And the overall the suppression function we can see here, roughly after nanosecond pulse, seems the suppressing function of the regular T cell decreased 50% roughly. We think this is very significant because if we decrease the regular T cell function, because we know other modalities such as radiation also be used to treat cancer-induced immune response, and it was reported radiation treat tumor actually promote T regular cells grow and also enhance their function. As you can see here, after radiation treatment, regular T cell function almost double increased. So we think this assay might show that the nanosecond pulse have some advantage over other tumor cancer treatment. So then, why those regular T cell decrease function? We go further, look at those called regulatory T cell function biomarker. Very important ones for WNBB and TGF beta look like they both, interestingly, after 4 hours, they have transiently increased those function markers, indicate they might also increase suppress function, but after 1 day, it's a very significant drop and of course, maintain the low level of those function markers. So we don't know what is the meaning, but because we know that at the end we have strong immunoprotection, also we suspect the transient increase of those function markers may not affect overall immune response. Then very importantly, we know the major cytotoxic T cell to kill the cancer is called tissue resident memory cells. So if you look at here, tissue memory resident cells, CD8 T resident memory cells, gradually and significantly go up after this 3 or 7 days nanosecond electrical treatment. So to summarize our discovery, after nanosecond electrical treatment, the multiple immunosuppressor cells, a significant drop include regular T cells, tumor-associated macrophages, and myeloid-derived suppressor cells. The reason they are dropped for the tumor-associated macrophage is probably the induced death by nanosecond pulse, and also the nanopulse seems selectively kill active regular T cells. nanosecond electrical pulse treatment also damage the suppressor function of the regular T cells. And we also observe the tumor-associated nanosecond electrical pulse treatment promote tumor-associated macrophage from M2 type to M1 type. And the nanosecond electrical pulse seems no damage to affect CD4 and CD8 T cells. And we also observed the cytotoxic T cell or CD8 tissue resident memory cells significantly go up. So I would like to acknowledge the major work done by our PhD student, Anthony Nananja, and also Megan, technician, and Brittany also help for cell culture and animal study. We also thank Dr. Bibi for his advice, and we also have engineer Edwin and Chinji help for the nanosecond pulsar. This study was supported by NCI and Pulse Bioscience and Virginia State Catalyst funding. Thank you for your attention.
Okay, so the SICKI presentation was not hello SICKI, you are here. Okay, thank you very much for the presentation, and thank you for fit very well the time. So there is any question from the audience for SICKI? So questions? Okay, so we are going to, there is one question. There is a question. It says that, did you notice any systemic immune response post-treatment?
Yes, so I will say this study most focus on the tumor microenvironment. So for systemic immune response, we have published in the Journal of the Cancer, International Journal of Cancer Research before. We observe the systemic change. We see all those suppressor cells, including regular T cells and myeloid deposits, significantly go down almost immediately after treatment. I think this is early response related to a tumor burden effect, because so depending on the tumor ablation, we have model if complete ablated tumor for the long follow-up, we know it. So they will be the most significant go down almost to the normal level, normal mice level. If we have a partial ablation, because we see the tumor not completely, some tumor grow back. Those mice we see actually between the middle. So look like those suppressor cells in the systemic is proportionally to the tumor burden. So that's why we more focus on this time, more focus on tumor microenvironment, because we think it's most important.
Okay. So no more questions. So we are going to move. We're going to move to the last speaker of this second part of the webinar. So the last talk is by Laure Gibaud. Laure is research at the Laboratoire des Interactions Moléculaires en Réactivité Chimique and Photochimique at the CNRS in Toulouse, in France. So her research interests focus on the use of tissue engineering, and more specifically on the in vitro reconstruction of human skin substitutes to understand the mechanisms of electroporation at the tissue scale. So Laure, the floor is yours.
Thank you, Javier. I'm going to share my screen. Is it okay for you?
Yes.
Yes.
Perfect.
Good. So I'm happy to be here to present a side project we have, which deals with cold atmospheric plasma and its effect on extracellular matrix remodeling at skin scale. So just to say a few words about cold atmospheric plasma, it's the fifth matter of it's the fifth state of the matter. It means that it transforms gases into ionized gases. And there is different components in the plasma, which are electromagnetic fields, heating, reactive oxygen species and nitrogen species, ions, UV light, and visible light. And since there is a lot of reactive species which are generating during plasma irradiation, there is some biomedical application I will not speak about, which are about sterilization and decontamination, as well as anti-tumor treatment. And there is some new topics about wound healing, which can be improved using cold atmospheric plasma. And most of the studies are led in vivo using murine models. And we can see an improvement of wound healing of skin. But there is also clinical trials which show that there is beneficial effects of plasma, cold atmospheric plasma, on chronic wound healing. And in this topic, we were looking at what happens at the extracellular matrix ECM scale, in particular in collagen, when exposed to plasma. So just a word about material and method. So here is the way we produce plasma. And you can see the plasma at the bottom of the pen. And we choose we decide to use a fixed distance between the plasma and the samples. So it's 2 centimeters. So just to let you see, on the bottom of the wells, there is dermal substitute. There is 600 microliters of cell culture medium on the top to cultivate the dermal substitute. And we expose them to the plasma for a few seconds. And we have 4 experimental conditions. So the control one, non-exposed to plasma. We use also the gas alone because this is a helium plasma jet, and you have to have a flux of helium and to put the electric field between electrodes to transform this helium in plasma. So we have the control condition with gas alone, and we focused on 2 times of duration of exposition, 2 minutes and 30 seconds. And just to show you what kind of model we use, it's a self-assembled human skin dermal substitute, which is produced by tissue engineering and the self-assembly approach, in which cells produce themselves their own extracellular matrix by stimulating them with ascorbic acid for a month. So the cells are able to produce extracellular matrix and to produce a 3-dimensional tissue. So in this case, it's a human dermal cells, so it's dermal substitute. And this extracellular matrix was characterized by this group, by proteomic, and they show that all the components of the native skin, human skin, are formed in this model. So it means it's a good model to study extracellular matrix. And just to show you how it looks like, here is a dermal substitute we will expose to the plasma. And here are some characterization of the model. So it's histological staining. In blue, you can see the collagens, and in red, the cells. Here is a second harmonic generation, which is produced by the fibrillar collagens. And these are electron microscopy to see the fibers of the collagen within the tissue. And here, the dermal fibroblast and all the extracellular matrix will embed them. So just to begin, we did some chemical analysis of the cell culture medium exposed to plasma, helium plasma. And we saw it's already known, but we were able to quantify the quantity of reactive oxygen and nitrogen species, depending on the duration of exposure to plasma. And we can see that the more we expose the liquid to plasma, and the more there is reactive oxygen and nitrogen species. So we choose this 2 duration of exposure to be sure that the cells within the tissue are not killed by plasma. So it's okay. So the cell viability is preserved. And then we focused on extracellular matrix remodeling. So first we looked at procollagen type 1 secretion in the supernatant over the time after treatment. So So in blue, it's a control. In purple, it's a gas alone condition. In red, it's 2-minute exposition to plasma. And in green, it's a 30-second exposition to plasma. And I will make a short story because nothing happens, both at the secretion level of collagen. We also quantified in the total of the tissue the quantity of collagen by quantifying hydroxyproline, which is a specific amino acid of collagen. And nothing happens in terms of quantity of collagen in the dermal substitute, even after 5 days of culture after treatment with plasma. We also looked at the MMP activities, which MMPs are metalloproteinases, which are known to degrade collagen in tissue. And there is no overactivity or decrease in activity of these proteases after exposure to plasma. And we looked at, by ELISA, we looked at the main MMP, which is MMP1 in the skin, and there is no modification in quantity of MMP over the time after plasma treatment. So it means that when you expose your skin substitute to plasma, it appears that there is no major remodeling in collagen content. But just to be sure, our device and our way to produce plasma was efficient. We checked for its anti-tumor activity because it's known that plasma-activated medium are able to kill in a certain way tumor cells. So we just to be sure, if we don't see any effect on ECM remodeling, it's not a matter of technical trouble. So we looked at spheroid, tumor spheroid, which are made with HCT116 colorectal cancer cells. And when they are exposed to plasma for 2 minutes, we can see 1 day after treatment a crown of detached cells, which are dying cells, as we can see by CAT-PAS3 and 7 activity in the tissue, as well as by using the propidium iodide as a fluorescence probe for plasma membrane integrity. And if we look at the growth curve of the spheroid after treatment with plasma, we can see that when they are exposed for 2 minutes to plasma, they have a growth delay. And if we measure the number of cells within the spheroids at this time point, so 4 days after treatment, we can see that when they are exposed to 2-minute plasma, there is a decrease in cell number. So in conclusion, it means that we are not able to observe any extracellular matrix remodeling after plasma treatment, but we know that this plasma is efficient because it keeps its anti-tumor activity on the tumor model. And just before finishing, a few words about electroporation, because it was the main topic we are looking for. And we were asking what about electroporation and extracellular matrix remodeling. So we hypothesized that the electrical component of electroporation without any drugs can modify cell behavior. So we test ECT parameters and gene electrotransfer parameters. And just to say, because it's not the topic today, but we did a huge and deep multi-method analysis of this answer of dermal substitute to electroporation, both with chemists, physicists, and biological analysis, especially by transcriptomic and ELISA. And our conclusion, which are coming soon in the Journal of Invasive Dermatology, is that when you apply electroporation on its own without any drugs on dermal tissue, you can see a lot of different results, which all goes in the same way. There is a decrease in synthesis of collagens. There is a decrease in the maturation of collagen, especially for these key enzymes. And there is an increase in degradation of the collagens by an increasing of MMP activities, which is reactive oxygen species dependent. And I hope I will be able to give you more information about this in the next year in the World Congress. So I just want to thank you for your attention, and Sarah, which did the main experiments of this presentation, as well as Nofel Merbahi for the plasma input, and Michel and Aurélie for the biological part. Thanks for your attention.
Thank you very much, Laure, for your presentation. Any question from the audience to Laure?
Yes.
It's a question for Siki. Okay, you can ask.
Yes, very interesting talk. So my question is related to your plasma condition. You see, it's weird. I think at the end you show the mechanism, the remodeling actually goes through ROS pathway, but the plasma supposedly generate ROS. So my question is, why plasma don't affect remodeling? Have you checked the condition? Because I saw when you do skin culture, you use vitamin C inside. Do you have any antioxidant actually cancel those ROS?
When we treat the samples, we are removing ascorbic acid, which is an antioxidant agent, you're right. And just to say, for the electroporation effect on remodeling of extracellular matrix, it's depending on ROS, but it's not ROS coming from the electroporation, but coming from the cells after the stress by electroporation. And when we quantify the reactive oxygen species, it's much higher in the plasma condition than in the electroporation condition. It means that we are not at the same range of oxidative stress. And maybe there is a threshold we don't have to pass through to get the cell response. I hope I answered your question.
So related to that, because I saw you when you treat skin, you have when you generate the skin, you use some vitamin C in the medium. Is that when you treat, you have some antioxidant in the medium?
Yeah, when I treat them, I remove the ascorbic acid, right?
Okay, thanks.
Okay, any more question, Laure? Okay, so no more questions. Thank you very much, Laure, for your presentation again. And we are going, with your presentation, we finish the second part of the webinar devoted to basic science. So we have a break of 10 minutes. So right now is 20 minutes to 5. So we will start at 4:50. Okay, 4:45. 4:45. Okay, we will start in 10 minutes. 4:45.
Okay. This is Pierre Jaïs. I have a presentation after that, I think. Is that right?
Yes, you are the last one. Yes.
And I'll have to be finished by 5. Is that okay?
Okay. How long is your presentation? How long is
it should be less than 15 minutes.
Okay. So you are going to start at 4:45 and you will finish at 5 o'clock.
Okay.
Okay, it's okay?
Yep. Thank you.
I think we can start again.
Okay. Hello, everybody, again. It's a big pleasure to introduce all of you the last speaker of today's seminar. He is Professor Pierre Jais. Pierre is professor at the University of Bordeaux in France, and he co-managed the Electrophysiology and Hair Modeling Institute and leads the research platform Music Equipment. His research is focused on improving knowledge and treatments of electrical dysfunctions of the hair. So Pierre, the ground is yours.
Thank you so much, Javier. So I'm going to share my screen. And I'm delighted to be here. I'm a great fan of electroporation, which I've been working with in the context of my collaboration, mainly with Farpulse, but also with Afera, two startup companies that have been using electroporation to manage cardiac arrhythmias. And so these are my disclosures, and you also see that none of these technologies are FDA approved, but Farpulse is CE marked now. And so the first thing I'd like to try to explain is that we're at the present time, our market and field is dominated by atrial fibrillation, which is a very common arrhythmia in human beings. And we treat those patients mainly with catheter ablation, which means that we introduce a catheter in the body and in the heart, and we create this type of lesions. They are induced by increase of temperature. We're burning the tissue. And I think that quite often, this is animal work, this vein here was isolated, electrically speaking, but you see that there are gaps between the core of lesions. It's filled with hemorrhage and edema, but everything is there for reconnection, which is one of the issues we have with thermal energies. So we've been trying in our field to develop single shot devices to isolate the pulmonary veins where the fibrillation is coming from. And those devices are working well. Some of them are using cryo, some of them are using radio frequency, but they failed to demonstrate any superiority over this point-by-point encirclement of the pulmonary veins. And this is a little bit surprising to me. In theory, they should be superior, these single-shot devices. They should allow for faster procedures, and this is observed with fewer reconnections, and this is still not clear. And in addition to that, we would like to have an energy that is less dependent on the contact between the catheter and the tissue, that produces more durable lesions. Ideally, we should have no recurrences in 90% of our patients. We are not quite there. It's closer to 70 or 80% at best. It should be safe with no collateral damage. We'll see that in greater details. And it should produce a limited amount of destruction of the cardiac tissue that we treat. And so when the industry approached the electroporation field, and this is not a surprise to you, there were many options and many recipes that in fact impacted both safety and efficacy. So this energy, as any electrical energy, I guess, can be delivered in unipolar or in bipolar mode. Can use monophasic or biphasic waveform. And all these parameters on the right of the slide, as you very well know, will impact both safety and efficacy. So it's a complex energy to manipulate, and yet it has bring so much hope to the field that every single vendor and industry in the field has repurposed some catheters for electroporation. It's really getting big in cardiology now. And so this is the far pulse catheter with the electrical field. That's pretty much the scale in purple here. So we're lucky here because the cardiac cells are very sensitive and will be destroyed without destroying the vascular smooth cells, the nerves, and more importantly, the esophagus. The esophagus that is right posterior to the left atrium, in contact with the left atrium, and which can be damaged by thermal energies, but which apparently is not with electroporation. And this is showing you a work we did in Bordeaux, where we've performed some electroporation in patients that were compared with other patients in whom pulmonary veins in the left atrium were isolated with either cryoablation or radiofrequency ablation, point-by-point method. And so when you look at the late gadolinium enhancement MRI right after the ablation procedure, you can see that the anterior aspect of the esophagus that is contoured with the dashed yellow line here is taking the gadolinium uptake, meaning that there is an esophageal lesion next to the atrial lesion. But with the electroporation, we never saw that, never. So it seems that really we have some tissue selectivity with electroporation, unlike what we have with thermal energies. And it's very important because what is at stake here is to avoid these fistulas within the left atrium and the esophagus. So here in this animal experiment, we delivered at the level of the inferior vena cava for anatomical reasons, but this is a complication that may happen in patients between the left atrium and the esophagus. It's rare. It's probably 1 per 5,000 or 3,000, but it's devastating when it happens, with 80% mortality and strokes that will leave sequela to the patients. And unlike what we have with radiofrequency in this animal model, we have no visible lesion either on the inferior vena cava or on the esophagus with electroporation. So this tissue selectivity is observed both in patients and in human models. And there's more to that. This is trained MRI, which in a way measures the atrial contraction. And so you see here that with pulse field ablation baseline, you have some degree of contraction that is the same with patients treated by thermal energies. Normal, this is baseline. And then if you look acutely right after the ablation, it's decreased whatever energy is used, but at 3 months, you have a complete recovery with pulse field ablation, but nothing with thermal ablation. This doesn't mean that the area that has been treated has recovered contractility. It does not, but we measured the contractility in the neighborhood directly adjacent to where the energy was delivered in those patients. And there is more when you do this MRI right after the ablation. In those patients with pulse field ablation, you see that you have a very homogeneous and intense late gadolinium uptake, which is the lesion we've created with electroporation. It corresponds to the isolation of the pulmonary veins. The pulmonary veins are these structures here that appear now in gray while they were normal before the isolation, normal being purple, which means high voltage of electrical activity. And what is interesting here is that 3 months after the ablation, we have a very significant reduction in the lesion with pulse field ablation, but no reconnection of the pulmonary veins. They are still completely electrically isolated, and this is the endpoint of the treatment for atrial fibrillation. And this, in fact, means that we have not damaged significantly the extracellular space. We just killed the cardiomyocytes, but the vasculature is still there and allows for a almost normal vascularization of the tissue, so that the gadolinium is not trapped in the lesion. And this is very different from what we have with thermal ablation. Acutely, with thermal ablation, we have this lesion that appears much more heterogeneous, with some areas that we call black holes, corresponding to either intratissular hemorrhage or to a complete absence of vascularization. And none of this is desirable, so it's a good thing we don't see them with electroporation. And at 3 months, we have pretty much the same persisting lesion, which means that we have produced some persistent change in the tissue nature itself, not just destroyed the cardiomyocytes. It seems from this data that the tissue specificity is consistently observed from animal experiments to AF ablation in patients with no esophageal lesions with pulse field ablation, compared to what we have with thermal energies, and this is a major advantage. There is a stunning around the pulmonary veins that is transient with PFA, on contrary to what is observed with thermal ablation, and fewer impacts on atrial function with PFA. And different type of lesion with PFA, we have no intramural hemorrhage or microvascular dysfunction, fewer acute edema, and more homogeneous and intense late guard enhancement. And we have the advantage with PFA that we have more durable lesions, 96% lesion durability at 3 months systematic reinvestigation. This is well over what we were used to with thermal energies. We have extended that to persistent atrial fibrillation ablation. It becomes a bit technical, but it works. This is and one in the US that my friend Vivek Reddy is leading. So I have very few doubts that we have many reasons to be very optimistic on the success of this energy, which is new in cardiology, and that has so many advantages listed here, but said before and mentioned before. I think that pulse field ablation will, with some optimized recipe and single shot device, will likely demonstrate superior efficacy in the context of a randomized comparison over the gold standard. This tissue selectivity will bring to the field a superior safety, and this is a major advantage for patients, of course, and also for healthcare system. And I think also that PFA will redefine the real success rate of pulmonary vein isolation, because for the first time we have highly durable lesions. I thank you so much for your attention.
Thank you very much, Pierre. I know that you have to leave, but perhaps you have time for a couple of questions that
yes,
are
please.
in the...
If there are any, yes.
Okay. Can you check the questions in the chat? Or...
Yes, I can try. Oh, okay. So in the field of PFA, it is consistently perpetuated that cardiomyocytes myocardium is much more sensitive to electroporation than other cells tissues. You also mentioned tissue selectivity in your talk. Where did you think this selectivity comes from? Is this selectivity on the cell or tissue level? These are great questions, Damian. I think you know more than I do on this on these issues. I honestly don't have good answers to that. This is something we want to study in depth in my research institute, and we want to launch a program that will also try to answer those questions. I don't think we have much in the literature, because what have been done is quite old and has been done with a very different type of waveform and recipe for electroporation. And so I really think that this question needs to be reinvestigated. But, you know, the proof is in the pudding, and well, both the animal experiments and the observations in patients are very consistently showing this tissue selectivity, and I'm delighted with that. Yeah, transient stunning, how long does it last? I don't know. I just know that between 2 hours from the ablation procedure and 3 months, it has recovered in the periphery of the lesion. How long does it take to recover? I have no idea. We would have to, you know, do several MRI to our patients, and this is, of course, not very convenient, and it's not going to happen. So somewhere between 2 hours and 3 months is the best I can say.
Okay, thank you very much, Per, for your presentation.
Thank you.
So we are going to close the webinar, and for closing the session, I'm going to pass the ground with my co-moderator, Giampiero Pataro. Okay, Giampiero.
Thank you, Javier. I would also just a few words to thank all the speakers for the very interesting and stimulating talk during this first webinar. And I think along with you, I would also like to thank all the attendants to this webinar. And remember you that tomorrow we have a second appointment with a second webinar devoted on oncology. And afterwards, you can also, after registration, also to participate to the General Assembly of the ISBT Society. And then, and that's all. I hope to see you very soon. And in particular, for sure, we will see next year in Copenhagen. Have a nice day, and thank you again. Bye-bye.